Hello everyone, my name is Rafsan. I'm a PhD student at the Department of Medicine at Lund University, and my talk is about a toolkit that we developed called EasyNER, and this is our approach to use artificial intelligence to read, quote unquote, biomedical research papers easily. So let's get straight into it. Biomedical data is abundant. From the research that we do at our universities or our institutions or the hospitals, we get many different kinds of biomedical data. For example, patient journals, scientific literature, or other types of text, and this is what we are focusing on. We also get bioinformatic databases and unstructured datasets such as images or audio. And we use these datasets to treat diseases, improve the quality of life of patients, develop new therapies, and improve overall public health. But biomedical data is also scattered and complex. For example, if you went to PubMed and searched which disease is closely associated with COVID, you might get over thousands of hits. And this is just for one query. And if you look at the first paper, it talks about diabetes and COVID-19. But intuitively, you might think that perhaps infection or lung-related diseases would be more closely associated with COVID-19. So your query may not always be accurate. But at the same time, you have here over 2,000 results. So you have to go through 2,000 papers to actually understand or detect what kind of diseases are associated with COVID. As of today, there are over 43 million research articles in PubMed. And I know this because I counted it myself. Reading all these millions of papers manually is pretty much impossible. So we need a new solution. This is like searching for multiple needles in multiple haystacks. But we do know one thing, that we're searching for biomedical entities. So our solution is, of course, since we are living in 2023, artificial intelligence. So what we did is we developed a natural language processing based pipeline where you input your research papers and in the end you get your domain specific entities out of it. So for example, if you have a text saying cell line X treated with chemical C showed improved survival in disease D expressing the gene G in species S. And using the EasyNER pipeline, you can get output as shown in the picture, cell line X, chemical C, gene D, disease D, and species S. So the idea behind developing this pipeline was to make biomedical text mining easier for researchers. A quick overview. We used fine-tuned BioBERT deep learning models for detection, and we also used site-spacey dictionary-based models. And of course, this whole pipeline is scalable. So if you have your own models that you want to use or integrate to the pipeline, it's very doable. This is, of course, for the advanced users, but we have left this option for you. As training data, we used 5 domain-specific Hunt or corpora, which include cell line, chemical, gene or protein, disease, and species. So as an example of the output, this is deployment of the disease model on the COVID-19 related text. This is the COVID-19 dataset, which includes over 1 million abstracts related to COVID-19. And these are the top 15 detected results, but this is not it. You actually get a large file of all the detected entities that are ranked based on their frequency. And here, as you see, infection is actually the top most occurring entity that we found from these over 1 million abstracts. We also have anxiety, cancer, and we also note that diabetes is present within the top 15. But also symptoms of COVID, for example, fever, pain, inflammation, hypertension, and such. So as an overview, these are two different uh, text collections. So on one side we have autophagy related articles that we received, that we extracted from PubMed. And here you see that different chemicals, cell lines, disease, proteins that are associated. For example, mTOR and TSC are commonly known genes for autophagy or autophagy associated genes. And also for COVID-19, you observe different cell line, oxygen and alcohol, um, um, different diseases, infections, and such. So the idea behind EasyNER is to make text mining and information extraction easier for medical researchers. And while developing this toolkit, we emphasize on making this as simple as possible. So anyone can use it in their laptops and read thousands of papers. For detailed overview, visit our GitHub page, github/iteslab/EasyNER, and here's a link to our paper, which is on archive. Thank you very much for listening.